All right, good morning, Science 10. Hope that you're all having a nice Wednesday so far. And yeah, so just some housekeeping for today. Reminder that we have a few assignments on the back burner. So lesson 1 to 3 practice problems is an assignment due today. Please complete that assignment before going forward, as we're going to kind of assume that you guys have a good foundation base on some of the topics that we've already discussed. Okay. So assignments on the back burner. Don't forget, we have our physics unit quiz that we're going to be writing next week when we get back on the Tuesday, so on the 25th. Okay, so don't forget to kind of start chipping away and preparing for that again. Yeah, we will have a review class on the Monday before we write the test, just to kind of go over some of the concepts and review them and revisit them. So we will have a review, but again, you guys are going to have to kind of start preparing on your own a little bit as we are learning from home, okay? So just keep that on the back burner. Okay, so let's start off with just kind of reviewing what we talked about yesterday. So we were talking about the organelles found in plant cells, organelles found in animal cells, those that are found in both, those that are not found in both. Okay, so remember, plant cells have so this is what animal cells do not have. These are unique to plant cells. So a cell wall, chloroplasts, and vacuoles. Okay, these are only found in plant cells. And then in animal cells, we do not have a cell wall, no chloroplasts, no vacuoles, and it does have centrioles. Okay, we will talk about the importance of the cell wall to plants in the last chapter, which we will cover in class. Okay, so make sure we know the similarities and differences between the plant and animal cells, and that you know the responsibility or the function of each organelle in a cell. So you're going to have to know what the nucleus does, what the ribosomes do, what the endoplasmic reticulum does, etc., etc. Okay, so that is probably the biggest learning point up to right now in this class. Okay, so make sure that you are able to recall the function of each organelle. Okay, yesterday we finished off with talking about the cell membrane. So this is what makes up the outside of the cell. It kind of protects it from the external environment. And this is done with a phospholipid bilayer. Okay, so remember, we have our phosphate group, and then we have our lipid tail. And that is on both sides. I'm just going to draw this one a little sideways here. Okay, this is why we have a phospholipid bilayer, because there are 2 layers of there. Remember, our phosphate groups are hydrophilic, so water loving, so they point to the outside, and the lipid tails are hydrophobic, so water hating, afraid of water, so they point towards the inside. Okay, so that's what a phospholipid bilayer is, and proteins are embedded throughout the phospholipid bilayer, and they are involved with transporting things in general across the cell membrane. Okay, they're kind of like the bouncers of the cell. Okay, so The cell membrane, the phospholipid bilayer, it maintains equilibrium within a cell. So equilibrium being kind of like the happy balance of ions and elements and chemicals inside of the cell. So it maintains equilibrium within a cell by selectively choosing which substances can get in or out of a cell. So we call this a semi-permeable membrane. If everything was able to make it through, we would call it a permeable membrane. So if everything can get through, we call it a permeable membrane. And then if nothing can get through, we call it an impermeable membrane. Okay. The substances that enter and leave the cell may be ions or molecules with unique chemical and physical properties. These could either be very important for a cell or for a body, or it could be a harmful chemical for the cell that it would want to keep out. And again, these images down here, these are our 4 major classes of biomolecules. So carbohydrates, lipids, proteins, and nucleic acids. Okay, these are all going to be regulated by the cell membrane. Okay, so we're going to talk about how particles behave now. Okay, so just generally speaking, particles are going to go from an area of high concentration to areas of low concentration. This is a process called diffusion. Okay, so if we have, if we look at our images on the right side of the screen here, if we have a concentrated lump of whatever, okay, given time, it is going to diffuse or dissolve into a solution so that its concentration is going to be equal throughout. So it'll be homogeneous, right? Going back to our science 10. So in our first Petri dish here, we have a heterogeneous solution because there are not equal concentrations throughout. But if we give it time, eventually that concentrated mass will diffuse and create a homogeneous solution where the concentration is equal throughout. So making iced tea, this might be a pretty familiar experience that you guys may have seen before. So of course, when we're making iced tea, we're going to put lots of sugar in it, right? Eventually, or at the start, all of it will sink to the bottom, but as we stir, right, the iced tea particles, so the iced tea like crystals and sugar crystals and stuff like that, are going to go from that kind of like the lump that we have in this Petri dish from the bottom of the glass, and eventually it's going to diffuse into the whole cup so that it is the same concentration throughout. Okay. A concentration gradient. So this is when there is a difference in concentration against or across a membrane. Okay, so if we look at our image on the bottom right hand corner of your screen, we will notice that in particular the case of diffusion here, we have more particles on the outer side of the membrane than we do on the inner side of the membrane. So because of diffusion rules, naturally the particles are going to want to transport across that membrane, so there is no more concentration gradient. Okay, things in nature always want to be at balance and not have concentration gradients. In cells, however, they kind of thrive off of concentration gradients. Okay, and this is why the plasma membrane is important to us, because they're going to regulate the concentration gradients of elements and chemicals and ions and stuff. Okay, so we have sodium potassium pumps throughout our body, and is what it does, it makes sure that we do not have too much sodium inside of our cells. Okay, too much sodium is quite damaging to cells, and it's not able to like fire or communicate regularly when there's an excess of sodium. So the sodium potassium pump is focused to eliminate excess sodium in the cell and bring back potassium into the cell, which is another really important signaling molecule. Okay, so the big takeaway from this slide, the definition of diffusion, particles go from an area of high concentration to areas of low concentration. That's diffusion. And then concentration gradient. This is when there's a difference in concentration across a membrane. And this is not exclusive to biology. We will use the term concentration gradient in multiple scientific fields. Okay. So we're just kind of going to elaborate a little bit more on concentration gradient. So there are 5 things we should talk about for concentration gradients. So number 1, it must involve different concentrations. Okay, so across some sort of a border or boundary. Number 2, in cells, different concentrations may be separated by a membrane. Okay, remember our phospholipid bilayer is a membrane, okay, and that is involved with creating a concentration gradient within the cell and what is going on outside of the cell. Concentration gradients involve molecules or ions of a single type, okay. Concentration gradients, different molecules or ions will move along their concentration gradients independently of one another, and concentration gradients will drive diffusion and osmosis. And we're going to talk about osmosis a little bit later on today. Okay, so how particles behave. So osmosis, this is an important term, so make sure you are familiar with this. Osmosis is the diffusion of water in particular across a cell membrane. Okay, so if we look at our images at the bottom of the slide here, we have a semi-permeable membrane, meaning that only some things can get through the membrane, not all things. Okay, in this case, water is the solutes are able to pass through that semi-permeable membrane. Okay, so pardon me. This occurs when there's a concentration gradient, so the solute molecules cannot pass through. Sorry, I got those mixed up. So if we look at image 1 here, so where the levels of water are equal, we note that we have a concentration gradient across this membrane here. We have more solute on the right-hand side. So naturally, water is going to move against their, so water is going to move into the right beaker because it wants to reduce the high concentration of the solute molecule in this beaker here. So there's going to be a net movement of water to the right in this particular case. Okay, so if we look at the beaker here, we will find that there's more water in the right-hand beaker because of the osmotic pressure of water. It wants to diffuse the high concentration of the solute in the right beaker there. So this should actually say here, water molecules move against their concentration gradient across the cell membrane. Okay. All right. So we can kind of predict the direction of water by comparing the solute concentrations across a membrane. So if it's hypertonic, so this is relative, this is comparing the solute concentration outside of the cell to the solute concentration inside of the cell. Okay, so in a hypertonic, we have a higher solute concentration outside of the cell because water wants to reduce that concentration gradient and kind of like bring it back to equilibrium. The water will leave the cell in order to attempt to reduce that concentration gradient. Okay, so that's hypertonic. Higher solute concentration outside of the cell. Next, we have isotonic. This is the same concentration of solutes across the membrane. So the solution outside of the cell and the intercellular chemical composition is equal. So there's not going to be any movement of water molecules across that membrane. Okay, hypotonic. This is when we have a high solute concentration inside of the cell. And because, again, the water wants to reduce that concentration gradient, that water is going to enter the cell. Okay. So an example of the movement of water in real life, if we eat a really high in salt meal, so maybe if we get like cheeseburgers and fries from McDonald's, has a lot of sodium in it. After we ingest that high sodium meal, we're going to have lots of sodium circulating in our bloodstream. Okay. So if we have lots of sodium in our bloodstream, more so than what is actually inside of our cell, what type of movement of water will this cell experience? If there is more solute concentration outside of the cell, then water is going to leave the cell. Okay, so if you eat a really salty meal, one reason that it dehydrates your body is because a lot of the water is leaving your cells in order to kind of neutralize the high sodium content within your bloodstream. Okay. So here is a short clip on how osmosis works. I'm just going to show it because it's quite helpful and it's a good summary.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules, such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
Excellent. So that's a really good summary there. I hope I don't get in trouble for posting that in this video. I think I'm okay because I'm not publishing this video for monetary purposes. It's for educational purposes. So I think I might be all right there. Okay. Let's see. Yeah, so let's actually stop there for today. We'll finish the rest of this lesson tomorrow. Okay. Now you guys have your topic 1 to 3 lesson worksheet to complete. So please take the time today to finish that. And as always, please let me know if you have any questions or if you need help with anything. I'd be more than happy to give you some assistance there, okay? So enjoy the rest of your day, and thanks again for tuning in for today's lesson. See you soon.